Hi, everyone. Our next speaker is Zhong, Assistant Professor of Computer Science at Princeton. Her group develops machine learning and computer vision methods for problems in cryo-electron microscopy with the goal of revealing how proteins move, interact, and carry out their functions inside of cells. Ellen received her PhD at MIT in 2022, where she created CryoDragon, a now seminal deep learning method for reconstructing heterogeneous 3D protein structures from noisy cryo-EM images. Her recent work includes new methods for heterogeneous reconstruction and in situ visual proteomics. Please join me in welcoming Professor Ellen.
Thank you, Ziming. And thank you to the organizers for their heroic efforts in putting together this workshop. I think it's a really great renegade NeurIPS workshop that, probably beats out a lot of the other NeurIPS workshops going on today. And it's also such an honor to be here on the other side after organizing for the last couple of years. And so it's great to pass the torch and kind of see what this field has turned into and all the excitement and for this opportunity to bring everyone together. Great. So yeah, as Ziming mentioned, my group works on developing AI and machine learning methods on various types of molecular machine learning problems with a focus on discovering new structures and new conformations. And we focus on a particular kind of imaging data called cryo-electron microscopy, or cryo-EM. So, fun fact, when we first started organizing MLSB, I think almost more than 5 years ago, this was like structural biology and machine learning approaches for kind of 3D protein structures felt like this niche area compared to genomics, compared to other topics. And so just a group of us got together and we're like, let's do a workshop on structural biology. And, it was like a complete surprise when the results of AlphaFold 2 at CASP was announced about a week prior to our workshop. And, since then, I think we're all familiar with why we're here. There's been what I like to think of as the broad liberation of 3D protein structure across all of the biosciences. And I think there's, many exciting things about what we can do now, both with structure prediction and then in complementary methods in de novo protein design. And, these days it's quite common that these unified sequence structure generative models are state of the art for all these problems in both structure prediction, design. But, on the scientific side, we're interested in understanding additional functions, and so the goalposts are yet advancing, and we're still extremely data limited for many problems, which is why I feel lucky that our group focuses on, active analysis of these interesting biophysical measurements that are extremely noisy to reveal these new protein structures from experimental data. And in particular, proteins and other biomolecules form these large dynamic macromolecular machines. And if we really want to understand, the function of these machines, ideally we would understand their entire conformational ensemble and their spatial contextualization. Oops. Unfortunately, techniques to get the data to study this are rather limited. So we have NMR, which I believe you heard about this morning, typically limited to small proteins, although you can get kind of specific measurements for much larger systems. What I like to call avant-garde crystallography-based approaches that look at some of the deformations when you shoot a crystalline lattice with strong lasers or magnetic pulses. Then there's, pure in silico or computational approaches for probing potential motions of these protein molecules. And so molecular dynamic simulations, I think there's a lot of really interesting, perhaps like emergent phenomenon from simulating these simple Newton's equations of motions and simple force fields. And I think a lot of the research that's super exciting going on here is, can we hack these unified kind of AlphaFold or other kinds of deep learning models to probe the specific kind of conformational states that might be present already in the data or interpolated from a lot of related structures. But what, sparks joy for me is, can we just observe? And I think that's one of the magical opportunities in cryo-electron microscopy. Let's just take images of these proteins and reconstruct what their ensemble is directly from experimental data. So in parallel to this huge revolution in structure prediction and ML for proteins, there's been completely in parallel this ongoing resolution revolution in cryo-EM, so non-deep learning based. received the 2017 Nobel Prize in Chemistry, and it's been particularly exciting since cryo-EM has opened up new areas of structural biology that, of these systems that previously couldn't be studied or crystallized. And this has been driven by breakthroughs in both the hardware and software of cryo-EM, so in particular direct electron detectors that fundamentally increase the amount of signal in the collected images so that we can now see things at atomic or near atomic resolution. And then concomitant with these hardware improvements, there's been a number of software improvements, both kind of new reconstruction algorithms, which I'll talk about today, GPU compute, and also significant automation and democratization of access to cryo-EM imaging. And so what I'll talk about today are some of the new computational challenges posed by cryo-EM and also new opportunities for this broader field of structure determination and what we can now reveal of proteins and biomolecules. All right, so for a bit of background, this is what I call the kind of different computational stages of cryo-EM image processing. The images that you get off are micrographs, and then there's various pre-processing steps, motion correction, particle picking. These are typically using kind of classical signal processing based algorithms. And then the focus definitely of our group is this 2D to 3D reconstruction problem. So can we process this data set of 2D cryo images? So here are real images from the SARS-CoV-2 spike protein, which was the structure that was determined about 1 month after the onset of the COVID-19 pandemic. Can we reveal the underlying 3D structure, 3D kind of cryo-EM density map from the data? Then, given a sufficiently high resolution density map, can we build in the atomic model to facilitate the final structure interpretation, feed into the next generation of the structure prediction or protein design models? And then now, so something that our group works on is revealing these dynamics from the data. And now we're in this era where we can reconstruct these complex ensembles. I think there's now this downstream problem of how do we analyze this distribution of structures. We have this abstract learned latent space. How do we make sense of it and actually describe the relevant modes to do the science of understanding how these molecules work? Okay, so cryo-EM reconstruction. This is a super interesting, challenging inverse problem. Experimentally, so the schematic on the right, we have a hopefully relatively pure solution of our molecule of interest, and it's flash frozen in a very thin layer of vitreous ice. And so these are our molecules. They're tumbling randomly in solution before being flash frozen. So they're captured from some unknown orientation. And we model it. So each image, yi, is modeled as a projection of this density volume, vi, projected at some pose that we don't know, phi I. This image is then convolved with the point spread function of the microscope, or the CTF, and then we finally model a bunch of additive Gaussian noise. So the goal is to estimate the volume and the poses, so these unknown viewing orientations, and this is typically done with maximum likelihood techniques. So in the standard approach for cryo-EM reconstruction, each individual image is a different volume VI, but we typically just estimate a single volume V. And this has been this resolution revolution, so there are many state-of-the-art software packages that do this, that solve this challenging inverse problem. And just to give you a high-level intuition on how these algorithms work, most of these methods are done in the Fourier domain, since we have the Fourier slice theorem, which states that the Fourier transform of a 2D projection of a volume is a central slice out of the 3D Fourier transform of the volume, where the orientation of the slice is perpendicular to the projection direction. And so this suggests this way of reconstruction, given some initial volume. So these methods typically operate with these iterative EM, or expectation maximization algorithms. Given some initial volume, if we have our images, we can align all of our images to the volume. And then given those alignments, we can convert them take their FFTs, convert them into the Fourier domain, insert them at kind of as planar slices in their relative orientations, build up our representation of our molecule in the 3D Fourier domain, and then take the inverse FFT to get back to our real space volume that we can now visualize. And this is like this iterative process that repeats until convergence. So that's how homogeneous cryo-EM reconstruction algorithms typically work. Then we have this so-called heterogeneity problem. And really, so, each image in cryo-EM, so these are real cryo-EM images, each image, which you can't really see anything, but you have to believe me that each contains a unique molecule in some unknown orientation, maybe at some unknown conformational state. And so the ability to image these heterogeneous structures is one of these major opportunities in cryo-EM, especially relative to X-ray crystallography, where we have to experimentally rigidify each copy of our protein in the exact same conformation or crystalline lattice. Here we actually get the experimental data needed to study this kind of intrinsic heterogeneity of our system. Unfortunately, you can imagine how this complicates this inverse problem of 3D reconstruction. And so the standard approaches for dealing with heterogeneous data include discarding or kind of ad hoc filtering based approaches. And then really the workhorse is something called 3D classification or multi-class reconstruction, where we model this image formation model as originating from a discrete mixture of K independent voxel arrays or K independent structures. And so a typical cryo-EM processing workflow might look something like this, where at the beginning you have millions of images that you've collected and particle picked from your electron microscope. And then through this, so these are created by humans, these kind of processing workflows, they're in the SI of any single cryo-EM paper. And through this user-driven ad hoc procedure where you pick K, you do this kind of iterative 3D reconstruction process. By the end, you have 2 atomic resolution structures, which is really exciting. But in this case, 90% of the images have been thrown away. And so the identification and analysis of heterogeneity, especially continuous forms, so can we model the kind of underlying continuous motions of these macromolecules, is an open problem. And just a quick shout out, I originally learned about this problem from reading this paper from 2 mathematicians at Princeton, Roy Letterman and Amit Singer. And the kind of main research question motivating CryoDragon, and continues to be a research question motivating the methods development in our group, is can we design modern machine learning methods that learn these complex, typically continuous distributions over data sets to this heterogeneity problem in cryo-EM. Okay, so this is the image formation model that I just talked about. For heterogeneous reconstruction, we extend this with this additional latent variable. So instead of a single volume, now we parameterize some class of functions that describe a set of volumes. And so one thing to keep in mind are what are the sources of heterogeneity that we might see in the images. So these molecules are intrinsically dynamic. We want to understand their dynamics to understand their function. But also the sample can contain multiple species. So I'm already getting kind of tongue-tied, but there's 2 different sources of heterogeneity, conformational and compositional. And so, typically I've been calling this conf-het and comp-het. And then there's also real kind of these artifacts and non-idealities during imaging that are not modeled by this simple image formation model. And these days, there's been a proliferation of different heterogeneous reconstruction algorithms. And so different methods typically use different choices of, this distribution of volumes, how we represent that, and also different methods for inference or optimization. So there's lots of different design choices. This is a review we wrote a couple years ago, back when there was like 12, or something around there, but now there's even more. So cryo-EM image processing is this very iterative procedure. And so in this heterogeneous case, both methods treat poses as known. So, you split up your reconstruction process, assume everything can be aligned, you infer the poses, and then you infer this residual heterogeneity of conformations or compositions. Okay, so the method that we developed is called CryoDragon. It's an acronym. It stands for Deep Reconstructing Generative Networks. And we frame kind of heterogeneous reconstruction as unsupervised learning of a deep generative model or a VAE of cryo-EM density maps that can be learned directly from the 2D images. And so the task here is, instead of reconstructing, a single structure, we now reconstruct this continuous distribution of structures. And something that's been particularly exciting is that CryoDragon's neural kind of representation, which is this coordinate-based neural field, has shown to be broadly applicable in other areas of computer vision, like kind of 3D or novel view synthesis. And so something kind of moving forward that we're interested in is establishing additional crosstalk between these fields. Okay, so where are we now? Something that's super exciting, especially at this intersection of both AI and experimental sciences, is thinking about how we can push the boundaries of heterogeneity. And so increasingly, cryo-EM and also cryo-ET has been used to study more and more complex or less pure samples. So already we can get these structures of these large dynamic complexes and their different conformations, discover like new states. But can we also, or this is a question, right? Can we also reconstruct complex mixtures that might have many different binders? Can we look at intact cells with the technology of in situ cryo-ET? And so one of the main motivations of our group is, can we develop methods? And I like to organize kind of two orthogonal directions, methods for achieving higher throughput and methods for revealing structures at higher resolution, ideally atomic resolution. Okay, and so, now CryoDragon, something that I was particularly motivated to do during my PhD was create a software tool so that people can actually train these deep learning models on their cryo-EM data to analyze their data sets and look at their structures. And so we have an open source software package, and I'm lucky to have a software engineer working in the group to help maintain and support this. And then we've also, so CryoDragon does not do everything. There's lots of limitations. And so a lot of the research directions right now are tackling some of these limitations. So pose estimation being one of them, so with CryoDragon AI, and kind of extensions into new modalities like cryo-ET with CryoDragon ET. And timing-wise, I don't know how I'm doing, but I'll briefly mention two vignettes. One is pushing resolution with atomic modeling and cryobolts, and then pushing throughput with CryoHive and CryoNet. Okay, so actually, I won't speak that much about this, but you can talk to Minkyu and Jeff. They have posters, and I believe there's an oral later today, but we tackle this problem of generalization in cryo-EM. And also, we look into methods that are models that are expressive for extreme compositional heterogeneity. So beyond like tens of structures, but to hundreds of structures or even 1,000 structures simultaneously. So please find them and chat about this. And then the last one thing I wanted to mention was that a reminder that in cryo-EM, we get these density volumes, but really what we want are dynamic atomic structures, right, from this experiment. And so I think something that I'm very excited about, and I think there's a lot of other works right now, can we leverage these priors from protein structure prediction or protein folding models in the structure determination inverse problem, right? And so protein structure prediction, they're fast, they encode all this prior information that we have on protein structures, but it doesn't predict dynamics or consider the context, and also requires validation. On the other hand, cryo-EM reconstruction, these are experimental measurements. We capture the dynamics or kind of the ensemble, but we currently do not leverage any prior information on protein chemistry or physics. And so you can also find Rish. Rish is somewhere here. And this was just presented at NeurIPS a couple days ago. But in cryobolts, we are using these protein or diffusion-based protein structure prediction models as a prior for this problem of atomic modeling in cryo-EM. So here, can we turn these, cryo-EM density maps, maybe lower resolution cryo-EM density maps, can we use that as a guidance term in these structure prediction models? And there's maybe two kind of complementary reasons why we would do this. So currently, structure prediction, there's the observed phenomenon that they're typically unimodal in terms of the structures that they produce. And so can we get them to produce the ensemble observed from experiment? And then on the cryo-EM side, there's the actual direct, super useful application of atomic modeling, right? And that is currently done in a fairly manual process. And so can we help do this, especially now with these generative models that can generate, or where you can sample an arbitrary number of density maps. Okay. The specific kind of contribution of cryobolts is a recipe for guidance that takes into account both the global shape of the protein of the density map, and then also the local features. You can take a look at our preprint, and then just one quick vignette on the results. So this is one transporter system. The real cryo-EM structures had four different states, and with cryobolts, we can accurately model all transporter states. So in contrast, structure kind of atomic modeling based approaches that don't integrate this prior information that operate in a purely bottom-up fashion produce highly incomplete models because of the low resolution of these maps. So I think there's some interesting questions now posed that this poses. how do we validate, right? So if the experimental data is now this low resolution structure, how much can we actually be sure that the atomic models that we build are supported by the data? And on the other hand, if we're using these structure prediction models as priors, how much does using that limit the ability to discover new conformations, new compositions, right, that whose like sequence we didn't put into the model? And so I think these are, extremely important questions for the field of both like cryo-EM, but also structural biology, and maybe even kind of AI for science in general. Okay, so the last part of this talk, I wanted to mention, or kind of, we're at this time where we can reconstruct these molecular movies. And these different algorithms, CryoDragon, 3DVA, 3DFlex, Dynamite, they use different sorts of priors in terms of the representations, in terms of how they optimize. And I think now it's in the realm of the user or the structural biologist to think about what is the biological question they want to answer. Do we want to find compet? Do we want to look at kind of conformational ensembles? And one of the challenges is that there's no ground truth, right? So we're trying to determine the structure. And right now, especially in the methods development realm, evaluation currently requires benchmarking by eye. And just as an example, this is an extremely noisy, homogeneous synthetic data set. Recovar is this probabilistic PCA-based approach. There's CryoDragon in the middle, and then 3DFlex is this deformation model of a tetrahedral mesh. And so there's no heterogeneity, but you can see that there's a lot of artifacts going on. And on the right, since this is a motion-based model, the noise in the data manifests as motions. So this is something to be extremely aware of, right, when we're trying to now develop these new methods for heterogeneous reconstruction. So one thing that we're doing is developing rigorous benchmarks. The heterogeneity problem is itself heterogeneous. So we've broken this up into both compet and compet, although you can definitely have the kind of composition of the two. And we've also broken things up into diagnostic or simple data sets that are easy to qualitatively evaluate, as well as challenging data sets that motivate new methods development. And so one of those is the Spike MD data set. The underlying data set contains 46,000 atomic models sampled from an MD simulation. This is the SARS-CoV-2 spike protein. And this is the results from 10 different heterogeneous reconstruction algorithms. We can see there's lots of different conformations that we reconstruct. And hopefully you can appreciate that all these methods, many of them have these low dimensional learned latent spaces, but these are arbitrary. How do you actually compare them? How do we actually quantify how well we're doing, especially for these super complex ensembles? So these are, we have some solutions, but these are still open problems for the field. And then the last thing is just a shout out for this new CARA challenge that we put together. So this was in collaboration with the Flatiron Institute and the CCPEM. It stands for the Critical Assessment of Heterogeneous Reconstruction Algorithms. And right now we have both synthetic and real data sets, and people should definitely try it out. There's some really interesting challenges in all of the different data sets in CARA. Cool. And so to end, just summarizing a couple of the frontiers of cryo-EM that can be enabled by ML. So I briefly mentioned heterogeneity. So can we push heterogeneity? Can we get extreme scale heterogeneity of complex mixtures? How do we analyze and the landscape of these different motions at hopefully atomic resolution, summarize those distributions? Something I haven't talked about, I don't have the time to talk about today, is the in situ cryo-ET work. So I'm just showing this cool movie on the right, just to you remember there's some cool stuff going on in situ. There's a question of what is the role of synthetic data in this regime? Can we train models on synthetic data that can generalize to real data sets? And what is, or can we develop methods and validate them on synthetic data, and will they still work on real data sets? Foundation models is a big question. So right now in this area of cryo-EM, it's pretty distinct from many other areas of machine learning where each model is specialized for a single data set. And currently it's extremely hard to generalize for many different reasons, one of which is the noise, the high amount of noise in the images. And so can we, build these foundation models? Why would we want to do that if we have this multi-million-dollar microscope and we're trying to see the atoms in the system? And when I talked about cryobolts, we're at this interesting interface between structure prediction and determination. If the structure is now just a hypothesis for some downstream design task, do we need to determine the structures, and how do we validate any structures that we determine, especially if the algorithms are leveraging all the prior information from these unified models? And then finally, the cryo-EM kind of image processing pipeline. Reconstruction is just one of many different tasks. It's an extremely iterative process or iterative pipeline. And so can we automate, can we have an agentic workflow to turn that kind of crazy flow chart that I described in the beginning to some black box system? Maybe. And with that, I'd like to thank the members of my group. A couple of them are here. This is us at the zoo the other day. And our collaborators, funding sources, and thank you all for listening.
We have time for some questions.
Hey, excellent talk. So I have a question about the cryobolts, especially about the performance. So I think the idea is very kind of interesting, but from what I'm seeing, it kind of depends heavily on the prior. Let's say the bolts are, alpha doesn't really generate a feasible structure, or it's generally like, a wrong structure, very different from the ground truth structure.
Yeah.
Well, these, what's a convergence rate of this kind of approach?
Yeah, that's a great question or a great point, which is that the cryobolts, like these methods that are guiding structure prediction models, it's not going to rescue kind of structures where the base model itself can't successfully kind of predict the structure. And so for a lot of these gigantic multi-protein complexes, if it doesn't have their orientation correct in the beginning, then it's not possible to rescue that with guidance, at least in our current framework. And so I think there's 2 points. One is that maybe it makes more sense for the specific targeted area of kind of fitting conformations and fitting the conformational distributions of a particular system. But hopefully, moving forward, as these structure prediction models are getting better and better, we can use them more as these priors in this atomic modeling problem.
Ellen, I have a question. I've been wanting to ask this actually for quite a while. So to my recollection, CryoDragon uses a VAE, so a variational autoencoder, and searches in the latent space to be able to fit this. How much of that do you think is just interpolation in what it thinks it learned versus actually learning physics-driven dynamics, as you call it, as in a movie, so to speak?
Yeah, so the movies, this is a great point. The movies that I'm showing are just kind of interpolations through the latent space. We're just doing a random walk through the latent space and generating structures to get this sequential movie. But that's just an arbitrary kind of path that we're taking. And I think there's lots of interesting things to do to connect that with actual molecular dynamics and kinetic pathways. But right now, we're just estimating the distribution from the data.
Thank you for the talk. This is kind of a tangential question. Do you think it will be possible to extend the guidance approach to data in the fluid space instead of real space, where a lot of the measurements of such as X-ray scattering takes place?
Mm-hmm. Yeah, I don't see any reason why you wouldn't be able to. So I think that's an interesting direction.
All right, let's thank Ellen again for the great talk.